Hello viewers, myself Palki Hazarika, a research scholar of the Department of Zoology, Guwahati University, and I am also working as a guest faculty in the Department of Zoology, Borooah College, Gauhati, Assam. In this presentation, I am going to discuss about various components of blood and their functions. But before going to start the presentation, I would like to mention that this presentation is a part of the initiative taken by the Zoological Society of Assam to help the undergraduate as well as postgraduate students in their studies during this period of COVID-19 outbreak. Before going to the detailed account of the presentation, I would like to mention the learning outcomes. After going through this presentation, the students will be able to learn the following. At first, a brief idea on what is blood along with some of the properties of blood. Then various components of blood, mainly of human blood. Then we will discuss about various functions of each component of blood. Then a brief idea on what is hematocrit or packed cell volume, then structure of hemoglobin and function of hemoglobin. Now let's take a look into what is blood. In this discussion, we will discuss about the vertebrate blood, especially of the human blood. Blood is a body fluid that circulates throughout the body of an individual by the pumping action of the heart to transport oxygen, carbon dioxide, nutrients, hormones, heat, waste, etc. It is also called as fluid connective tissue because it has a fluid matrix called plasma, and since it connects all parts of the body, it is called as connective tissue. Blood occupies about 8% of the total body mass in an adult human. That is, the total circulating volume is about 5600 milliliter in a 70 kg man. It is slightly alkaline in nature and the pH of blood ranges from 7.35 to 7.45 in vertebrates. In case of human, the blood pH is approximately 7.40. The blood is red in color in all vertebrates due to the presence of a respiratory pigment, hemoglobin. The branch of science deals with the study of blood, blood-forming tissues, and the disorders associated with them is called hematology. The word hemo or hemato is used in blood-related study and it comes from the Greek word aqua or hemia, which means blood. The blood that flows through the blood vessels is known as whole blood and it comprises of two major components. The fluid matrix called the plasma, which occupies about 55% of the total blood, and the formed elements, which occupies about 45% of the total blood. Now let's discuss about the various components of blood in detail. As mentioned earlier, the blood comprises of two major components, that is plasma and formed elements. The plasma again comprises of water as well as solutes. The solutes may be inorganic and it contains sodium, chloride, magnesium, potassium, bicarbonate, biphosphate ion, etc. Whereas the organic solutes of the blood plasma comprises of proteins, lipids, carbohydrates, hormones, vitamins, amino acids, etc. Three major proteins are found in the blood plasma, which are albumin, globulin, and fibrinogen. On the other hand, three types of formed elements are found in the blood, that is erythrocyte or the red blood corpuscles, leukocytes or the white blood corpuscles, which can be again divided into granulocytes and agranulocytes, and the third type of formed element found in the blood is the thrombocytes or platelets. Let's discuss about blood plasma in detail. The fluid matrix of blood is called plasma. When formed elements are removed from the blood, it gives us plasma. Plasma can be separated from the blood by means of centrifugation technique. As shown in the figure, as a result of centrifugation of blood, the formed element settles down at the bottom of the centrifuge tube and a straw-colored liquid remains as a supernatant. The resulting supernatant is called the plasma. It contains approximately 91.5% water and 8.5% solutes, of which 1% comprises of salts or ions, and remaining 7.5% contains proteins, nutrients, vitamins, hormones, carbohydrates, lipids, amino acids, urea, uric acid, etc. Functions of the major components of plasma are as follows. Water helps in maintenance of blood volume as well as transportation of molecules. Whereas plasma proteins play a major role in maintenance of osmotic pressure and pH of blood. Three major plasma proteins are found in the blood plasma. Number 1. Albumin. Albumin maintains the colloid osmotic pressure of the blood, which is important in maintaining a balance between water inside the blood and that in the tissue fluid around the cells. It also acts as binding and carrier protein. Number 2. Globulin. Three types of globulin proteins are present, that is alpha, beta and gamma globulin. The gamma globulin contains high density lipoproteins which act as carrier of fats to the cells for energy metabolism. Whereas the beta globulins contain low density proteins and helps in transportation of fat into the cells for the synthesis of steroids and cell membrane. The beta globulin also stimulates cholesterol plaque formation, which is considered as a risk factor for arterial and heart diseases. And the gamma globulins or antibodies are called immunoglobulin, and these are produced by a subset of immune cells called B lymphocytes or B cells. The gamma globulins are responsible for body's humoral immune response. They recognize pathogens via specific receptors. Third protein is the fibrinogen. It is the precursor molecule of the clotting protein fibrin, which traps platelet to form blood clot. The complement proteins are another type of plasma proteins involved in immune and inflammatory reactions in response to a variety of infectious particles. Besides the functions of plasma proteins mentioned in the previous slides, these plasma proteins can also perform some other functions. For example, they keep the blood pH slightly alkaline by binding excess hydrogen ions in the blood, and they can also supply amino acids if required by being broken down by macrophages. The third major component of the blood plasma are the ions or salts, of which sodium is the most abundant ion carried in the plasma and contributes most of the plasma osmolality. Besides sodium, calcium, potassium, chloride and bicarbonate are also found in the blood plasma. Calcium also helps in muscle contraction, whereas sodium, chloride and potassium helps in transmission of nerve impulse. On the other hand, the bicarbonate ions regulate the pH of blood. Other components of the blood plasma are nutrients, vitamins and hormones. Nutrients like glucose, fats, amino acid, mineral, etc. and vitamins are served as food for cells of the body, whereas the hormone helps in reproduction and sexual differentiation and growth and development of an individual, maintenance of internal environment as well as regulation of metabolism and nutrient supply of an organism. Formed elements. Let's take a look into various formed elements of blood and their functions. Formed elements of the blood comprises of two cell types, that is erythrocyte or red blood cells and leukocytes or white blood cells, and cell fragments, that is thrombocytes or platelets. The process of formation and development of formed elements from the hematopoietic stem cells is called hematopoiesis or hemopoiesis. Hematopoiesis starts before birth in the yolk sac of an embryo and later in the liver, spleen, thymus and lymph nodes of a fetus. Bone marrow is the primary site of hematopoiesis in the last 3 months before birth, and it remains as the main source of blood cells after birth and throughout life. Next, erythrocytes or red blood cells. As shown in the figure, mature RBCs or erythrocytes are biconcave in shape, bearing a diameter of about 7.8 micrometer and a thickness of about 2.5 micrometer at the thickest point and about 1 micrometer or less in the center. Unlike many other cells, RBCs do not have nucleus and hence RBCs can easily change their shape, which help them fit through the various blood vessels of our body. However, while the lack of a nucleus makes a RBC more flexible, it also limits the lifespan of the cell as it travels through the smallest duct or blood vessels, damaging the cell's membranes and depleting its energy supplies. The lifespan of a RBC is about 120 days. RBCs are the most abundant cells in the blood, accounting about 40 to 45% of its volume. A healthy man usually have approximately 52 lakhs RBC per milliliter cube of blood, and it is approximately 47 lakhs RBC per milliliter cube of blood in case of a healthy woman. The percentage of total blood volume occupied by RBC is called hematocrit or packed cell volume. As RBCs have the ability to concentrate hemoglobin in the cell fluid, anemia and reduced oxygen carrying capacity can be assessed by measuring the hematocrit. Hematocrit should be 40 to 45% in healthy adult males and 38 to 46% in healthy adult females. Therefore, a low hematocrit reflects a low number of circulating RBCs and it is an indicator of decrease in the oxygen carrying capacity. Whereas, a high hematocrit may reflect an absolute increase in the number of RBCs or a decrease in plasma volume. The process of formation and development of erythrocytes or RBCs is known as erythropoiesis. The yolk sac is the site of primitive nucleated RBC production during early embryonic life. Then, the liver becomes the site of RBC production during the middle trimester of gestation. The spleen and lymph nodes also produce RBCs during this developmental period. But the bone marrow becomes the main source of RBC production during the last month or so of gestation and after birth, and it remains up to the age of 20 years. After that, most RBCs continues to be produced in the marrow of the membranous bones such as the vertebra, sternum, ribs, and ilia. Functions of red blood cells. An important function of RBCs is to carry oxygen from the lungs to the tissues, and oxygen in turn helps in cellular respiration. RBC contains a huge amount of carbonic anhydrase enzyme that helps in transportation of carbon dioxide in the form of bicarbonate ion from the tissues to the lungs. The carbon dioxide in turn helps in regulation of blood pH and influences the affinity of hemoglobin for carrying oxygen. RBCs are also responsible for most of the acid-base buffering power of whole blood, as they contain an acid-base buffer called hemoglobin. Next is the structure of hemoglobin. The hemoglobin is the principal protein inside the RBC, which accounts for 1/3 of the mass of an RBC. The presence of hemoglobin in the RBCs allows the blood to transport 30 to 100 times as much oxygen as could be transported in the form of dissolved oxygen in water of blood. As mentioned in the previous slide, it is also act as an acid-base buffer during the conversion of carbon dioxide into bicarbonate. The hemoglobin found in the adult human being is hemoglobin A, whose molecular weight is 64,458 gram per mole. It is composed of two components, apoprotein and a prosthetic group. The apoprotein found in the hemoglobin is called globin, which consists of 4 polypeptide chains, and depending on number and sequence of amino acid residues, the chains are designated as alpha chain and beta chain. 2 alpha chains are present in the globin, and each one is composed of 141 amino acid residues, whereas 2 beta chains are present in the globin and each one is composed of 146 amino acid residues. The second component of hemoglobin is the prosthetic group. It is an iron protoporphyrin 9 complex called heme. It is a globular protein, water soluble, and it is quaternary in structure, and it helps in oxygen transportation. It is a planar molecule and it has tetrapyrrole structure composed of 4 pyrrole rings. In this figure, the 4 pyrrole rings are designated as A, B, C and D. Each pyrrole ring is a 5-membered ring made up of 4 carbon atoms and 1 nitrogen atom. And all the 4 pyrrole rings are interlinked with methine bridges. These bridges are labeled as alpha, beta, gamma and delta, respectively. The inner carbon atoms of pyrrole rings are linked to methine bridges. The hemoglobin is called a metalloprotein as it contains ferrous ions as prosthetic moiety. It is also called as chromoprotein due to the presence of a pigmented prosthetic moiety. Functions of hemoglobin. Hemoglobin helps in transportation of oxygen from pulmonary alveoli to body tissues. It transports carbon dioxide in the form of bicarbonate ion from body tissues to pulmonary alveoli. Hemoglobin also acts as protein buffer, which is an important buffer of plasma. The second type of form elements is the leukocytes or white blood cells. Let's take a look. WBCs or leukocytes are usually oval-shaped and varies in size as they are of different types. Unlike RBCs, leukocytes possess nuclei and lack hemoglobin. Normally, the number of WBCs in human blood is 5,000 to 10,000 per cubic milliliter of blood, which accounts for about 1% of the total blood volume of the body. White blood corpuscles are primarily produced in the bone marrow and derived from the undifferentiated hematopoietic stem cells, whereas the maturation of WBCs takes place in the thymus. Normally, two types of WBCs are present in the blood of a human: granulocytes and agranulocytes. The granulocytes are again divided into three types. Number 1, polymorphonuclear neutrophils, which occupy approximately 62.0% of the total WBC. Number 2, polymorphonuclear eosinophils, which occupy 2.3% of the total WBC. And number 3, polymorphonuclear basophils, which occupy approximately 0.4% of the total WBC. The agranulocytes, on the other hand, are divided into two types: monocytes and lymphocytes. The monocytes occupy only 5.3% of the total WBC, whereas the lymphocytes occupy approximately 30.0% of the total WBC. In this section, we will discuss about different types of granulocytes in detail. Number 1, polymorphonuclear neutrophil. The neutrophils are the most abundant type of WBCs, which occupy 54 to 62% of the total WBC. Neutrophils possess multi-lobe nuclei. The main function of neutrophils is phagocytosis, which means cellular ingestion of the offending agent. Phagocytes must be selective of the material that is phagocytized, otherwise normal cells and structures might be ingested. On approaching a particle like bacteria or any other foreign substances to be phagocytized, the neutrophils first attaches itself to the particle and then projects pseudopodia in all directions around the particle. The pseudopodia meet one another on the opposite side and fuse, and they form an enclosed chamber containing the foreign particle. Then the chamber invaginates to the inside of the cytoplasmic cavity and breaks away from the outer cell membrane to form a free-floating phagocytic vesicle inside the cytoplasm. A single active neutrophil can usually phagocytize 3 to 20 bacteria. And the neutrophils provide second line of defense. Second type of granulocyte is the eosinophil. Only 1% to 2% of the total WBC is occupied by the eosinophils, and the nucleus is bilobed in shape. Eosinophils are weak phagocytes and they exhibit chemotaxis. But in comparison with that of neutrophil, it is doubtful that the eosinophils are significant in protecting against the usual type of infection. However, eosinophils are often produced in large quantities in people with parasitic infection. Eosinophils attach themselves to the juvenile form of parasites and kill them by releasing hydrolytic enzymes, by releasing highly reactive forms of oxygen, or by releasing some larvicidal major basic proteins. Third type of granulocyte is polymorphonuclear basophil. Basophils occupy less than 1% of the total WBC and they possess lobular nucleolus. Basophils play an important role in some types of allergic reactions because antibody that causes allergic reactions, that is the immunoglobulin E, has a special tendency to become attached to the basophils as well as to the mast cells. Then when specific antigen reacts with the IgE antibody, an antigen-antibody complex forms which results in rupture of the basophil, and this basophil releases large quantities of histamine, smaller quantities of bradykinin, serotonin, etc. Basophils also secrete an anticoagulant called heparin and a number of lysosomal enzymes. And these substances cause local vascular and tissue reactions that causes allergic manifestations. The second type of leukocyte is the agranulocyte, which is again divided into two types, the monocyte and lymphocyte. Monocytes occupy to 9% of the total WBC, and they possess bean-shaped nucleus. Along with neutrophils, the mature monocyte, that is the tissue macrophages, attack and destroy foreign invading agents by means of phagocytosis, and these monocytes provide first line of defense. Next is lymphocyte. Lymphocyte occupies about 25% to 33% of the total WBC, and they possess disc-shaped nucleus. After birth, lymphocytes are found in the bone marrow as well as in the lymph nodes, thymus and spleen. Two types of lymphocytes are found, that is B lymphocyte and T lymphocytes. B lymphocytes are again divided into plasma cells and memory B cells, whereas the T lymphocytes are divided into helper T cells, cytotoxic T cells and suppressor T cells. B lymphocytes. The plasma B cells of the B lymphocytes secrete large quantities of antibodies, while the memory B cells are formed after primary infection and exhibit antibody-mediated immune response. Number 2. T lymphocytes or T cell. Of the three types of T cells, the helper T cells are again divided into two subtypes, Th1 and Th2. The Th1 cells secrete interleukin-2 and gamma interferon and are related to cellular immunity, while the Th2 type of helper T cells secrete interleukin-4 and interleukin-5, and they interact primarily with B cells or B lymphocytes in relation to humoral immunity. The third type of T cells, that is the cytotoxic T cells or killer cells, destroy transplanted and other foreign cells, even some of the body's own cells, with their development aided and directed by helper T cells. Whereas the suppressor T cells are capable of suppressing the functions of both killer cells and helper T cells. These cells can prevent the cytotoxic cells from causing excessive immune reactions, which might be damaging to the body's own tissue. Here we have come to the third type of formed element of blood, that is the thrombocytes or platelets. Let's take a look. Thrombocytes or platelets are small granulated bodies that aggregate at the sites of vascular injury. They lack nuclei and are 2 to 4 micrometer in diameter. Approximately 1,30,000 to 4 lakh per cubic milliliter of blood is occupied by the cell fragments platelets. The platelets are produced in the bone marrow by the megakaryocytic cells, which spins off bits of cytoplasm and extrude 60 to 70% platelets into the circulation and the remaining amount into the spleen. Functions of platelets. Platelets secrete vasoconstrictors which constrict blood vessels, causing vascular spasm in broken vessels. They also form temporary platelet plaques to stop bleeding. That means they help in blood clotting. Platelets secrete procoagulants to promote blood clotting, dissolve blood clots when they are no longer needed. That is, platelets help in hemostasis. This means the process of forming clots in the walls of damaged blood vessels and preventing blood loss while maintaining blood in a fluid state within the vascular system. Blood coagulation or blood clotting is a series of chemical reactions that culminate in the formation of fibrin threads. Blood clotting involves various substances known as clotting factors. These clotting factors include calcium ion, several inactive enzymes which are synthesized by the liver cells, various molecules associated with platelets or released by damaged tissues. Vitamin K is a fat-soluble vitamin that is needed for the post-translational modification of certain proteins required for blood coagulation. It is involved in the carboxylation of glutamate residues in proteins to form gamma-carboxyglutamate residues. As shown in the flow chart, platelets and damaged tissue cells release prothrombin activator, which acts on prothrombin in the presence of calcium ions to produce thrombin. Thrombin acts on fibrinogen in the presence of calcium ions to form fibrin threads. So this is all about the various components of blood and their functions. All the information included in this presentation were collected from certain books and research papers. Hereby I have given certain references and suggested readings. These are the names of books and research papers the students can go through for more information. Hereby I have given my email ID hazarika.palki@gmail.com. For any query, please contact me at this email ID. Thank you.